This is the preparation for Lecture 21. Lecture 21 is not going to be covered on the second midterm. So if you want to minimize your prep of this material, that's fine. You can decide for yourself when you want to do it. We're going to be continuing on this topic of setting up the body axes, the germ layers, and so on. In a little bit more detail. So we covered the beginning parts, the just basic sort of structures and the simple description of movements during gastrulation for the frog and the zebrafish in lecture 18. And this is where we're going to layer on a little bit more detail. We're going to look at fate maps and just a few of the genes that are responsible for the setting up of the body axes. So just a reminder that the animal-vegetal axis is set up during oogenesis, and it's temporary. It's just at the beginning part of development. Animal-vegetal axis doesn't have any real relevance to the body of these animals once they've fully developed. A reminder that yolk is concentrated on the vegetal side, and it's very dense. It's an impediment to cell division. That means when the cell is starting to divide, the cytoplasm is up here. The nucleus is probably sort of around here of that first single cell. And then as the divisions proceed, they go really quickly through the top part because this is just cytoplasm to be divided, but then it goes very slowly through the yolk. Okay. The first two cleavages are along the animal-vegetal axis, so this way and then that way at right angles to each other. And then the third one basically, divides in an asymmetric way with smaller cells up top and bigger cells at the bottom. And this is just a recap of what we covered in the prep for lecture 18. So if you do some molecular work and you do some staining, you will find that there are a couple of genes that are found at the vegetal pole. So the fact that this was called the vegetal pole when people were first studying it meant that they thought nothing was really happening here, that all the action was up in the animal pole. So this is kind of an outline of the embryo, the fertilized egg really. Not very well drawn because this is actually the edge. God. And so, and here's where the nucleus is. And then there is a protein called VEG-T, which is a transcription factor. And it's found sort of around the egg, but there's a strong localization of it right at the vegetal pole. And this is a really important factor. And VEG1, which is a signaling molecule from the TGF-beta family, also is localized here. There are a couple of others, but the two important ones to remember for now are these two, and these are maternally provided messenger RNAs, and they're localized here. And they are part of the signaling system that ends up setting up the body axes. Okay, so fertilization is actually the beginning of the dorsal ventral and also anterior posterior, as we'll see at towards the end of the lecture, specification. So the sperm can enter anywhere around the animal pole, and when it does, there is a reaction which is called cortical rotation. So the sperm enters at some position, and then there is a process by which dorsalizing factors, think about VEG1 and VEG-T, and they're localized mostly in the cortex, most concentrated at the vegetal pole. And sperm entry, think back to the nematode. And the fact that when the sperm entered, it triggered a reaction by microtubules that set up the whole localization of the PAR proteins to the posterior and others to the anterior. If you go back and think about that, this is another process by which the sperm enters, and that triggers a reaction too by microtubules. And what happens is they orient sort of away from the sperm, so that any molecules that are on this is in the cortex. The cortex is the cytoplasm just below the egg membrane, so it's kind of around the outer surface, and it's not a different structure than the rest of the cytoplasm, but there are materials localized there, and the cytoskeleton, the microtubules, are localized there too. And so through a shifting of those microtubules, the dorsalizing factors, which we'll talk about what they are in a minute, they are moved up and around towards the dorsal side. And it is because of the reaction of the microtubules to the entry of the sperm that everything lines up the way it does, and that causes these factors to move to a new position in the egg, and this will end up being where gastrulation initiates. Okay, so it's a very important process. There's a few slides on it, and a few more in the lecture than what I'm covering here in the intro. So the rotation is about 30 degrees, and it leads to a physical structure because when the cortex moves, it takes with it the pigment that's sort of underlying the membrane, and so we get what is called the gray crescent. So if you were to look at, and there's a picture coming up, if you look at the embryo, you actually see the formation of this gray. So what's happened is there's a little bit of pigment in this region, but also the yolk is kind of shifted upwards a little bit. So you get this different color, a physical thing that can be seen. So this gray crescent appearing was observed far before they knew what the reason was for it, but they thought that something was moving or relocalizing, and we now know that it is dorsalizing factors. And these are the ones: disheveled protein, Wnt11, messenger RNA, and Wnt5a. There's tons of different Wnts. A lot of these are also called X-Wnt because X is for Xenopus, just to specify which organism it's from. Okay, so again, the sperm enters, the microtubules kind of rearrange themselves, and they orient in a certain direction. It causes materials that were in this vegetal pole, including these three molecules, to move up to the future dorsal side, which is almost directly across from where the sperm entered in the first place. And this is just shows like the sperm has entered, and it's brought its centrosome with it, and that's what leads to this sort of orientation of the microtubules, and they orient kind of away from the point of sperm entry. And because they're all lined up together, then that shows how you can move materials in particular directions. When all the microtubules have the same orientation, then motor proteins can just kind of walk along in this direction, or the microtubules themselves can move right along the cortex. So how do we know that these microtubules matter? Like we can show that they line up in response to the sperm entry, but what is that proof? Not necessarily, but there are experimental manipulations you can do to try to deal with that. So if you treat the cells with cold shock, or you use a nicotazole treatment, both of these interfere with functioning of the microtubules. So the microtubules polymerize from units of tubulin, and either of these treatments prevent them from forming. When you do that, you don't see cortical rotation. And the eggs that result, they start going through cleavage, cell division and stuff, but the proper axes never form. They end up just taking on a cylindrical shape, the embryos. They don't have any dorsal structure, so you don't get dorsal specification. Why? Because you didn't have cortical rotation, and it didn't move the materials to the correct location and start the right signals and so on. And so they kind of form, a tube of vegetal structures, which is gut, basically. And when the yolk runs out, there's no more nutrition and they end up not surviving. And then there's some other evidence. So think about these mutations in terms of loss of function. So preventing cortical rotation is like a loss of function mutation because the thing that's supposed to be happening isn't happening. And then ask yourself, how could we do some kind of gain of function? Because when we're trying to find out what the role is, you want to do both of these things if you can. So what can we make it rotate further than normal? What happens? Does the dorsal pole form in a different place? What if you allow rotation to happen and then you induce more rotation to happen? What happens again? So these are two different ways of generating what we would call gain of function. So what happens? Okay, so here's a sort of a summary slide. So if you allow the rotation and just leave the embryo, it develops normally. If you inhibit the rotation with UV irradiation or cold shock or nicotazole, so there are multiple treatments that all lead to the same thing, which is the rotation doesn't occur, and so you have this tube of gut, this ventralized embryo. You can enhance microtubule polymerization, that is, make the polymerization more than normal. You do it by treating with D2O, which is heavy water. This is a hydrogen atom with an extra neutron, I think, in it. And so when you treat it that way, for some reason, it causes enhanced microtubule polymerization, and you end up with a dorsalized embryo. So it has lots of it has an eye, kind of an eye. There's overdeveloped mouth related structures. It's all kind of epidermis and neural cells, and this is dorsalized. It lacks a gut, and it lacks a lot of other structures. And then finally, you allow cortical rotation to occur normally, and then you put the embryos in a centrifuge, and you do this very slow centrifugation. They have to be, I think they fix them in place so that the rotation forces the rotation in the centrifuge forces the rotation in the embryo in a certain direction, if I read that correctly. And what happens is you end up with two different axes. So you have the first one that occurs from the normal procedure, but then if you wait a little bit and rotate again and move the dorsalizing factors a little further along, then you get two sets of dorsalizing factors and you get these conjoined twins. So we've done loss of function and two different ways of getting a gain of function, and we've seen results that suggest a very important role for those microtubules in dorsal specification. And here's even yet another one, which is lithium treatment. Now, lithium treatment, this is the experiment when I was talking about lithium treatment for sea urchin embryos, and they had vegetalized embryos. But when we treat a frog embryo with lithium chloride, we get a dorsalized embryo, so hyperdorsal. And remember what lithium treatment does. What kind of signaling is it mimicking? And then this is just a repeat of the previous slide that just shows that if you inhibit the cortical rotation with UV treatment, then you get the ventralized embryo. So remember that lithium seems to be an inhibitor of GSK3. GSK3 is required as part of this destruction complex and the destruction of beta-catenin so that it's not able to enter the nucleus. It's usually only when Wnt signaling occurs that the disheveled protein binds with the activated receptor and prevents the degradation of beta-catenin by the destruction complex and allows it to go into the nucleus and transcribe. In this case, it would be dorsal specific genes, right? Because it's dorsalizing factor. And so if we if we treat with lithium, that means that we're getting wingless signaling over most of the embryo. And so now all of it is dorsal because it's not confining the dorsal specification to one place. It's specifying dorsal everywhere, and we get this defective embryo. So when we're not treating with lithium, when we move those dorsal specific factors, the Wnt and disheveled protein, we lead to the accumulation of beta-catenin in the nuclei on the future dorsal side that will allow for activation of dorsal specific genes. We're only going to talk about one of the genes that gets transcribed in this region this time. And in this exact region, this is where the Spemann organizer develops and where gastrulation will begin later on. So Peter Newcoop was a famous embryologist, and he did a really famous experiment. He divided a 4-cell stage embryo in such a way that he divided the dorsal and ventral halves. And when you take the ventral half, you get a ventralized embryo, mostly gut structures. And the dorsalized embryo, interestingly, is close to normal. It's lacking the most ventral structures, so it doesn't have like a gut and some other structures, but it looks much more normal. And the key difference, well, many things, but one thing is that the dorsal half contains the Newcoop center, which is what organizes gastrulation and the development of a body axis and so on. So the Newcoop center is part of signaling, and he did some other kinds of experiments that showed what the Newcoop center actually was. And the Newcoop center, as I said, overlaps with the gray crescent, and it's actually it's a place where signaling molecules actually signal to the cells above them, and the cells above them are going to form the Spemann organizer. So there's 2 layers of organization. One is the Newcoop center, and then that causes the Spemann organizer to form, and then that organizer directs the gastrulation process. So this is the timeline. So the microtubule activity rearranges after fertilization, and the X-Wnt 11 and 5A, X, remember, for Xenopus, and Wnt, it's another one of these wingless ones, they are relocated as the cortex, the inner cytoplasm, rotates around 30 degrees towards the dorsal side. And then on the dorsal side, in the cells here, you will get beta-catenin in the nucleus. And then the Newcoop center is the region where you have lots of the beta-catenin in the nuclei, and it begins the expression of the gene CMY. And VEGT is part of one of the transcription factors that also that induces CMY, but also some other genes that we won't talk about. I'm trying to remember, but in a lecture or two we'll be talking about those. CMY is a homeobox transcription factor, and it has a number of target genes that are necessary for dorsal specification and other things we'll talk about later. So some other work that has been done with the Newcoop center is that you can do some experiments where you take cytoplasm from cells in the general area of the Newcoop center in one embryo, and if you inject them onto the ventral side of another embryo, you now have 2 Newcoop centers, and they specify for the cells above them to initiate gastrulation, and we wind up with these twinned embryos. You can transplant the cells as well, so either the cells or the cytoplasm will give you that effect. And it turns out that if you don't transplant cytoplasm or cells, but you just inject beta-catenin into these ventral vegetal cells, you get the exact same result as if you had transplanted the Newcoop center cytoplasm. So it is the beta-catenin in those cells that are part of this process, right? You can make it happen by just putting the beta-catenin in there. So if you might want to just review a little bit about the Spemann organizer experiment from chapter 1 and just remind yourself how that worked. So the anterior-posterior axis is really interesting because it turns out that the blastopore will end up being almost exactly the posterior pole. So what's funny is that both the dorsal pole and the posterior pole are set kind of at the blastopore. And so that doesn't make sense, right? But what happens is as the cells are invaginating, they're sort of going inside and rearranging, then the anterior, posterior, and the dorsal ventral kind of resolve into proper axes that are at approximate right angles to each other. So we start this way with the dorsal, kind of the dorsal pole at the dorsal lip and the posterior pole right at the blastopore, practically the same. But then as these cells are moving in, the dorsal pole kind of moves along, and so we get this shift, and they are practically at right angles with each other now. And we'll talk about there are some signals and things that we'll talk about. The cells that move in early are going to be anterior, so they get anterior specification, and when they reach this region up here, they kind of quote unquote know that they're supposed to form, anterior structures. And the signals they receive as they're coming through later on will give posterior specification. And so we have to talk a little bit about some of those signals. But for now, this is where we're going to leave that part. Now, if you look at the zebrafish, the formation is similar. Now, we don't know what the original symmetry breaking event is. We know that it is not sperm entry, but we don't know exactly what it is. Is it maternally laid down factors, or is it something else? Unclear. But what is known, here we're looking, so this is the yolk, and so we're looking down on the top from the animal pole when there's just a few cells, and it turns out on the dorsal side there's messenger RNA for a signaling molecule called nodal related 1. And it's thought that it acts along with beta-catenin for the specification of dorsal fates. So, and these are mRNAs, so we know these are maternally provided. And it might be just as simple as that. It's just provided, and it's just provided in a particular location. And so therefore, after cell division, there's going to be a couple of cells that are going to form the dorsal side, and they contain the nodal related proteins. Or sorry, messenger RNAs, but they're going to be translated into proteins. Okay. So if we look on the zebrafish, there are microtubule-based movements. So microtubules are set up, and certain dorsal determinants are actually transported. So we don't see what you would call cortical rotation, but we do see the transport of those proteins and messenger RNAs up towards the dorsal. We do see nuclear localized beta-catenin on the future dorsal side. And then in the shield region, just before it forms, really, this isn't really the shield, this is still the sphere stage, but this is the dorsal side, so this is where that shield is going to form. There's a lot of nuclear beta-catenin, and there is a gene called DARMA being expressed there. CMY is the homeobox transcription factor in the nucleus center of the frog, and DARMA is the equivalent gene. It's a homeobox transcription factor, but it's not a homolog, so they're not exactly the same, but they're the same kind of gene doing pretty much the same job in the two different organisms. So DARMA is required for correct development of the dorsal side of the fish embryo. Okay, and so this is the fate map of, so that's just that. I think I said in lecture 18 that I was only going to touch on sort of the similarity between the fish and the frog and just point out the differences as a comparison point rather than going in as much detail on the fish as we do on the frog. And I think I'll continue with that. It seems like a good way to do it. So this is a side view of the Xenopus fate map. And this is dorsal ventral, animal vegetal, because we don't have anterior posterior yet until after gastrulation. But if you look on the vegetal pole, that's where the endoderm is found. The mesoderm is in a sort of middle position here, and the ectoderm is on the animal half. The dorsal ectoderm is going to form the nervous tissue, and the ventral ectoderm and I guess the animal, sort of the central part, is going to form the epidermis. And then if we turn the embryo and look at it from the dorsal side, this just shows you kind of how it's organized. So your endoderm is here. Your mesoderm on the most dorsal part, that's going to be the notochord, which is going to be that tube of not tube, but rod of mesoderm that lies below the developing neural tube. The somites, which I talked briefly about previously, and we'll have a whole lecture on sort of somite development and so on, is in the more lateral place. And then on the ventral side is things like the blood vessels and the blood cells themselves, as well as kidney and some other internal organs. Yeah, that's everything. And then if you look at the zebrafish embryo for comparison, it looks really, really similar. So on the dorsal side, the mesoderm is kind of halfway down, notochord, somites, blood, kidney, heart, that kind of thing, the internal organs. Endoderm is in here, but it's mixed with mesoderm, so there are some muscle and other types of mesoderm that can develop from here. Down at the bottom here, this is a giant cell that is just yolk, and it's not divided. And then on the dorsal side is ectoderm, and on the more ventral side is the epidermis. So the fate map looks really, really similar, even though the embryos at this stage don't look as similar. So if we go back to the frog, even if you take late blastula and divide them, it turns out that if you divide them such that if you get them right through the Spemann organizer, the two halves can actually still form small, fairly functional embryos, which is really interesting because this is quite advanced for that to be happening. The key is, though, the plane of bisection. It has to be done so that there's enough tissue here to induce gastrulation and rearrangement and the formation of the body axis and all of that. If you were to divide on another axis, you would not get anything like that, right? So if you were to try to divide sort of dorsal ventral, you get only partial embryonic development in both cases. I want to finish off with a little bit about the signals that give rise to ectoderm and endoderm and mesoderm. In the early embryo, the early blastoderm, this part here is referred to as the animal cap. It hasn't got any contact with the marginal zone or the endodermal cells. And at the beginning, the ectoderm and the endoderm are specified particularly by factors, maternally deposited RNAs that get translated on fertilization. That should sound quite familiar because that's what happens in the fly. For example, maternally deposited messenger RNAs that don't get translated until after fertilization. But it turns out that the mesoderm is actually specified by a series of signals sort of between the ectoderm and the endoderm, and it'll be sort of this region in here that is going to give rise to the mesoderm. Prior to that specification, so if you're in the early blastula stage, you can excise the animal cap and culture it by itself, and you will get epidermal cells, so you get the ciliated ectoderm. And you can culture vegetal cells and you will get endoderm, so gut-like tissue, and there won't be any mesoderm. However, if you culture the two together, they will form mesoderm between them, and we'll be talking about that a little bit more when we do a subsequent lecture in a little bit. So these are the signals, though, and interactions to know about, and we'll talk more about those in the next lecture. So here's our early blastoderm, and so on the animal side, FOX1 FOX1E is expressed, and it specifies ectodermal derivatives. So it's telling the cells that that the signal is telling these guys to form ectoderm. There is another protein being expressed called ectodermin, and it's basically going, don't form mesoderm. So it's saying to these cells, you're not allowed to be mesoderm, right? So you've got an activating of ectoderm and a repressing of mesoderm. On the vegetal side, you have veg-T, which is really important. We'll talk a lot about veg-T. And nodals, where you have more nodals and then fewer across from the dorsal to the ventral. But veg-T specifies endoderm-specific fates and activates nodals. And the nodals are specifying mesoderm fates and repressing the expression of FOXE. So I want you to just think carefully about this figure, because the arrows don't show where these are going. They indicate interactions. So you've got your nodals being expressed in this region, and they're preventing FOX1E from being expressed there, so that because FOX1E is going to be telling the cells to form ectoderm. So we can't have it being expressed. So the nodals are going, you form mesoderm and don't express any FOX1E, because that's going to be confusing for all of us, right? For all of us cells. And then FOX1E is being expressed in here, and so is ectodermin, because the ectodermin is telling these guys, okay, there's nodal signals right beside you, but you're gonna form ectoderm, right? So the balance between this repression and this repression is what sets the line. So everybody on this side of the line is getting more of the ectodermin expression that shuts off mesodermal fates, and the cells on this side are getting more of the nodal that says yes, yes, form mesoderm, and very weak or no signals from ectodermin. So hopefully that makes sense. And then the same down here, these guys are endoderm cells, and then the cells up here are expressing nodals, which are, again, going to tell the cells in between to form mesoderm. So it's just 1, 2, 3, 4 signals to remember. And it is the fact that you've got maternally provided signals for ectoderm, maternally provided signals for endoderm, and then it is the interactions between them that set aside a layer of cells in between that, due to the signals they've received, then form mesoderm. And that's where that fate map comes from. And just to remember that nuclear beta-catenin and CMY are in this region, and they are contributing in some ways to the, what kinds of fates the cells can take on, and of course the initiation of gastrulation, and we'll talk more about that next time. Okay, hopefully that was